Okay, when you zoom into the leaves, sorry for that. When you zoom into the leaves, you are able to see that the xylem continues from the stem up to the leaves via the midrib and the veins which are in the leaves. Because I think you know that some leaves have parallel leaf veins, network leaf veins. Now, this is a continuation of the xylem within the midrib. Now, you find that these veins in the leaf, veins, I mean those ones in the leaf, are surrounded by what we call parenchyma cells, which you can call them mesophyll cells. In the middle here, there are some palisade mesophyll cells up here, as well as the spongy mesophyll cells down here. Okay, so you must understand that anatomy when you're looking at it. How does it occur? Now, according to this theory, effective evaporation of water from the mesophyll cells here, I think you can see in this picture, as water is being evaporated, it is facilitated, it facilitates the cohesion and tension forces here in the xylem, which push water up the plant. But then how does a loss of water in the stomata facilitate uptake of water via these forces? How does it facilitate the forces of cohesion and adhesion forces pushing the water up? How does it actually happen? Now you find that when water evaporates from the leaf surfaces here, where you're seeing my cursor dancing here, you find that these cells around, are you seeing these cells around? They are going to have a low water potential because they have lost water. So there is a low water potential here. Now, when these cells have a low water potential, it means they are less concentrated. Water is going to be drawn from the xylem to enter these cells where there is low water potential by osmosis. Now, when water enters here, it is being pulled, okay, from the xylem to enter these cells. And remember, these cells are continuously losing water by evaporation, okay? To understand it more again, you know that xylem vessels are always full of water. And when water suddenly leaves them, for example, water has just left them to enter these surrounding mesophyll cells, there's going to be a tension that is created or it is set up within this long column of water, this one here. Tension. What do I mean by tension? The word tension, it just means that force which tends to pull different objects apart. I'm assuming you're having a string here. So a force which is going to be pulling towards the, let's say, the right, and then towards the left. Let me do like that. Yes, towards the left. That's tension. Tension is a force which tends to pull two parts apart. Now, the moment water has been lost here, it's going to be pulling. It's a force that is pulling. That's the tension that I'm talking about. Now, the tension is transmitted up to the roots by the strong cohesion forces. Those are the forces that are in between xylem. And then those high cohesive forces between water molecules and high tension that set up in the xylem vessels brings about the upward movement of the water, upward movement of the water in a tall tree. So that is the summary of cohesion tension forces or cohesion tension theory or the transpiration pool theory. It is normally examined in paper 2 of biology 30/2, paper 2, which has those essay questions. That's where you can find such questions being asked. Now, this is the summary. So what has just happened, from what we have been discussing, is that water moves across the root soil down a concentration gradient. This is down a concentration gradient. So it's going to be moving down the concentration via the apoplast and the simplest pathways. So you still remember the apoplast and the simplest pathway. This picture, for some of you, are a little bit lost, is summarizing the cohesion-tension theory that tries to explain how water moves up this the tall tree. Okay. So we are beginning from down here. So after that, you got cohesive forces between water molecules which within the xylem. Then water is pulled up the xylem, and then what actually happens in the leaves, this transpiration actually happening. So water evaporates from the spongy mesophyll cells and diffuses into the atmosphere. When it has diffused, that one lowers the water potential in the leaf cells. Now, when water potential is lowered in the leaf cells, water is going to again move down the concentration gradient. So from the xylem, it is just going to enter into the mesophyll cells surrounding the xylem. And why? Because there's a tension at the top of the xylem, which is being created as transpiration occurs on the mesophyll cells. Okay. Then there's also some other force which you can talk about, which is capillarity. Now, capillarity is not a big thing. It's not so much special. You just need to know that it just means upward movement of any fluid as long as it is in a narrow tube. So since the water rises up through narrow leaves, it is also facilitated by capillarity through the stem. And this is because the xylem vessels are too narrow and the flow of water is maintained without breaking by both cohesive and adhesive forces. So capillarity is brought about by the xylem being narrow. If you want to understand capillarity more, think about how you suck soda. Why is it that if you are, I'm assuming this is the bottle. Okay, let me draw it here. This is the bottle of soda, and you're going to be sucking soda. Why is it that those straws that you use, I'm assuming this is your nose and that's your mouth, and you are sucking soda. So why is it that the straws are made when they are narrow? It is to increase capillarity. Capillarity is a force that aids movement of fluids within a narrow tube. Okay. Imagine if the straw was so huge, how would you suck it up? So if you have understood, there is this question which says a column of water in the xylem vessels of a tall tree ascends without breaking mainly due to, what do you think is the answer? A column of water in the xylem vessel of a tall tree ascends without breaking mainly due to. If you have an answer, you can type it here in the dialog box, whether it is A or it is B or C or D. You can always type it here and we see your answer. Mildred Tranila says it is B, cohesive force. What about the rest? Sandra says it is C. For Sandra, she has chosen C. Tranila, she said it is B. And Kesa Sarah says it is B. You need to, what about the rest? What have you seen? I don't know whether some of you have just not understood all, but I would expect everyone here to be able to answer. Emerald says B, that is cohesive force. So most people are choosing B and C, so, but I'm seeing now B is taking on cohesive force. We're looking at a force whereby a column of water in the xylem vessel of a tall tree ascends. Ascending means to move upward. Descending means to move downward, but ascending means moving upward. So a tall tree ascends without breaking, mainly due to. Okay, I think we can go with B. Cohesive force, because cohesive forces, those are forces between the water molecules themselves. Like if you have a drop here and another drop of water, they tend to pull one another. There is a force in between them that tends to pull to put them together. Then adhesive forces, those are the forces between the walls of the xylem and then the water molecules. Okay. Now those adhesive forces are going to prevent water from falling back in the xylem. It doesn't drop back. Just like you have sucked soda within the straw and then it falls back. Now those are the adhesive forces. Then root pressure, I talked about it last time, okay, which is generated mostly because of presence of the Casparian strip within the endodermis. In the endodermis. And then transpirational pull, we have just talked about it. So we can continue. Now, if you have understood, can you be able to explain transpirational pull by yourself? Can you? Now, because such a question can be examined in paper 2. And this is the summary which you must know of what actually happens. So if you can't, then you better revise that area harder. Now, this is a summary of transpirational pool, some of the of the plant, movement of water of the plant. So we shall begin with the cohesion tension force or theory. These are the bullets, the major factors whereby you can even add flesh on them in case the question comes in paper 2. The first one is water evaporates from the leaves because we're looking at the transpirational pole. And when water has been lost from the leaf, it's going to create a diffusion pressure gradient between the air and the xylem. And then a tension, that is a force which tries to pull. A tension exists in the water column extending from the roots to the leaves. And then adhesion and cohesion forces keep the column continuous, whereby adhesion forces don't allow it to flow back down. Cohesion forces, they're holding the water molecules up, so it's going to aid in movement of water up the plant. Now, that water is continually being pulled upwards as water evaporates from the leaves. What you must also never forget is capillarity, about spontaneous movement of water up narrow tubes. That is it. Just movement in narrow tubes. You can't speak of capillarity and you don't talk of narrow tubes. That will be out. And you know that the lumen of the xylem vessels and tracheids are narrow. Now, we are a little bit later, we're going to understand what xylem vessels and tracheids are. Then also root pressure, which is generated as a result of having a Casparian strip in the endodermis. Now, normally you find that the cell sap of root hair orays, the cell sap of root hair orays has a low water potential compared to the soil water solution. And then another thing is that water enters soil hair, root hairs by osmosis, and then continuous osmosis causes pressure forcing water a short distance up the stem. That is the root pressure. Now we can go to to the xylem and water transport. Now, and the next slides, we are going to understand the structure of the xylem. What is the xylem? Okay. What is it composed of? How is it? What is its nature? Where is it located? So you must Okay, I was still telling you, sorry for that, that to describe is to paint a picture of something in words. Okay, so let us begin with the xylem. Now, the xylem is the main tissue in the roots, stems, and leaves. And it's tissue responsible for, sorry for that unstable network a little bit. So it is the main, actually, let me underline this one, main tissue responsible for water transports in the root, stem, and in the leaves. Now, xylem tissue, what consists of mainly the xylem vessel and then tracheids as the main transporting cells. Okay, both are made up of dead cells. So that is what you must know. In most cases, when we speak of the xylem, we mean xylem vessel and the tracheids. Okay, both of these are going to be found in the stem, roots, and as well as in the leaves. So it doesn't matter. It's just like I'm assuming if you are having two cups, and each cup you use it, maybe both cups can do the same work, but they could be modified differently. Maybe one cup is plastic and the other cup is, let's say, melamine cup or metallic cup. It is the same thing. So they have what we call xylem vessel and the tracheids. Now, some books, even if you don't add this word xylem and you just call it vessel element, actually some books call it a vessel element instead of xylem vessel. They just call it vessel element, and then the tracheid. Now, the water transport takes place through the hollow lumen of these dead vessels and tracheids. I've already told you that sometimes they're called vessel elements. Let me write here the word element. Vessel element, and the tracheids. These are the ones which conduct water. And they are of two different types, as we are going to see. There is this one, you can see, this is how it looks like, the vessel element, and then the tracheid. But now, here it had been cut transversely to see inside, but here in picture one, it's not, this is how it looks inside, okay? And then you can see that this tracheid, it is kind of pointed on one side. So this is how it would look like. It tapers up, and this one does not taper. Can I please record work on my PC? There is no problem to record. You can always record. Then, xylem vessels have open ends, which offer less resistance. Let me first note this. They have open end. So this is the vessel element here in picture one. You can see it's like it's open-ended. Are you seeing here anything? It's just a boundary from one vessel element to another, but there's nothing like a barrier. It's just open-ended in between here. So things just go up and down like that. There's nothing like a barrier. But check, for the tracheids, they have their endings are not open-ended. They have pits. They have pits which connect to this. Like in Uganda, it's called obtuli. They have pits which can connect from one tracheid to another, like that. And so let me first get a question here, but I'm not seeing anything, sir. Now, you Kishu is not seeing. Try to first leave and then rejoin again. Telling me to ask permission from the host. I'm not the major host. You try to contact Bernard. Cells. Someone had a question here. There is no one. Okay. So xylem tissue. Tissue is made up of 4 cell types that function together. Remember, a tissue refers to a group of cells from your senior 1 biology. With a tissue, it's a group of cells that can perform a particular function. So, but xylem is made up of different tissues. The first tissue is the tracheid. The tracheid comes in different forms, as we shall see. Another one is the vessel element. These 2 cells, tracheid and vessel elements, are the major water conducting tissues in the leaves, in the stem, and in the root. And how do you describe a tracheid? A tracheid is going to be long, it will be narrow, and tapered and tapering. What do I mean by tapering? Something which tapers means that it keeps on narrowing towards one end. Now, this part here is tapering. So if it were like of equal size all through, like in the case of the vessel element, now this one is not tapering at one end, but this one is tapering. This is what we mean by tapering. With pits means that there are holes that join from one. I'm assuming there's another one which connects here up like that. But this one is open-ended. From one, you go to another, just like combining pipes, which are hollow. A vessel element is this one. One is the tracheid, like that. It tapers at one end. Vessel elements are large with thickened cell walls and no end plates. There are no end plates here in between. Things can just flow freely, but here there are end plates. Then secondly, apart from the water conducting tissue, there are also some other tissues. For example, the xylem parenchyma. Now, these cells are the ones which can be modified to form more of these xylem and of the tracheid and vessel elements. Parenchyma cells, they're just within there. Now, picture A is a longitudinal section through the stem showing the xylem vessels. Picture B is showing from above, that is a transverse section. This is what you normally see in the microscope. And this is what you normally see after staining. You will see these things. I don't know whether any one of you has had that chance to stain. Normally we use fluoroglucinol when you want to stain. There are some other stains like methyl blue. There are many kinds of stains that we normally use in the lab to stain those tissues. Then thirdly, the sclerenchyma tissue. Now this one is made up of fibers and sclerides. Sclerides even have what they call stone cells. Now those fibers are strong. They give support within the places where they are, within the stem or in the leaves, just for support. Okay, I can give an illustration of this using concrete. Concrete means enkokoto, when people are laying concrete to build a storage building, kalina. When they're building a storage, when they're constructing a storage building, they will have a column, and in that column they will put iron bars. Those iron bars can be reinforced with okay, those are the iron bars which they put there. They are tied together, and then they can put there a mixture of stones with cement and sand. You see, cement and sand to make that column of a storage building. So likewise, to make the thing strong. So likewise, within the stem, I'm assuming the iron bars are, because you could have like 3 different types of iron bars within the column. So imagine that some bars are tracheids and some bars are vessel elements, okay? And then probably the stones are acting as the parenchyma, xylem, and then you have the sand and cement acting as the sclerenchyma cells, something like that. So that's the kind of arrangement. To me, saying what is the use of parenchyma? I've told you parenchyma, they are cells which are normally thin. If you asked it broadly, let me first answer it broadly. Parenchyma cells are just simple plant cells, and they are found in almost every part of a plant organ, where their main job is to act as packing tissue. Packing tissue is that they fill up spaces within plants, and they can store even what food. They can store food. For example, if you want to see parenchyma cells in detail, just get an Irish potato, make a thin slice of it, observe in a microscope. You can stain with iodine solution. If you stain with iodine solution, you will be able to see some starch grains in it. So it's made up of very, very thin cell walls, and it can easily be modified into another as cells are differentiating. Let me first get another question here. J. Josh, unmute and say your question.
Excuse me, teacher.
Yeah.
Teacher, what about here? I have that the parenchyma cells have their ray parenchyma and also fusiform parenchyma. So which is which? Should we talk about that or we shouldn't talk about it?
Okay. Thank you for that question. Okay, ray initials, those things you study them under the section which we study when we are actually during the development of the xylem. I think that's where you've opened, isn't it so, Josh?
Yes, teacher.
Under development, yes. So when you're looking at development of the xylem, you are trying to see how is the xylem formed, okay? Now, when you're seeing how the xylem is formed, of course, you begin from, just like when a human being is being formed, you come from someone who is young. Now, the parenchyma cells are like the ones which are young and they have not yet specialized to perform a particular function. Like you cannot say now that this parenchyma is going to grow and become one which is going to be conducting water. It can be modified differently and it doesn't conduct water, and instead it becomes part of the ones which are just giving support, just parenchyma fibers. Okay, so I don't want you to at it in terms of that ray. First, leave that section of development of the skeleton of the xylem. First, look at the tissue itself after it has been formed. That's where I should open. I hope you're getting me now, Josh.
Yes, teacher, but teacher, I'm asking.
Yes.
Aren't we in histology?
Thank you for that question, Josh. Now, that's why I've told you that here in transport, we don't look at formation of the xylem or even the phloem. That is histology. That's where you study histology, okay? Yes. But here in a transport, we study these tissues after they have been formed, okay? So we study their structures and then how they conduct water. So right now we are looking at the structure of the xylem. Is that clear?
Yes, teacher.
Yeah, but of course some people decide to skip it, but even if you consult very many notes, you have to talk about this structure. Of course, some people cover it under histology. I hope you've got it.
Yes, teacher.
Okay. Thank you for that, Josh. So after knowing that we're having the trachea and vessel elements, so actually that can be an advantage that we may just even rush so that we don't waste time here in your understanding this tissue in case you covered it under histology. Now, most of the xylem tissue is made up of tracheids and vessel elements, which are both types of water conducting cells. So here I will make you aware of them, and I want to assume that you guys, some of you covered this under histology, so I will not overwaste time on it. Now, xylem vessels Of course, they have different thickenings, and lignin is laid down in various patterns. Okay, just like some of you in your houses, you can decide to paint different patterns within your walls, within yeah. So now this is lignin. Lignin is that structure which is reinforcing these cells. Now we have what we call annular thickenings. Okay, lignin laid out in circles, like in this picture here. This is annular thickening. Now some of you may be wondering, but what are those things, Banaye? Or what are they? These things are highly examinable. And the point in case, I have a slide which I'm going to show you. It was question number 4 of 2016 UNEB. 2006 thickenings. So some people think that these things are not examinable. I'll show you the question. That UNEB question of 2016, paper 2. And then we also have what we call spiral thickening, which is lignin laid in spiral. Okay. And then 3, the reticulate, or sometimes people separate them, but because they almost look alike. So reticulate forms some kind of network, network thickenings, and then scalariform thickenings, which is normally laid in ladders. Okay, and then you can see here the reticulate, and then the pitted. Now I got this picture from Functional Approach by MBV Roberts. This is what you'll find in Functional Approach. But when you open some other box, they have that pitted thickening. Let me try to draw it here mechanically. This is it. And then it will have those pits. Pits are like obtuli, like that. That is the one which is called pitted. Pitted thickening, like that. Now, xylem tracheids are similar to xylem vessels, except that the ends are tapered, okay, and cross walls are always present. So even the tracheids have them, okay, as well as the vessel end. But the difference is that the tracheids are tapering, like in this case here. You can see it's pointed on one side. Clarissa says that, how do I answer this question? Explain the distribution of mechanical tissue in plants. Okay, we shall talk about that. I think you've seen Clarissa's question. Now this question is normally taught under histology. If you have done histology, then you can ask such a question. But now we're looking at transport, because with okay. So this question is histology. I don't know whether they've done histology or not. So xylem vessels and xylem tracheids are highly specialized. Okay, let me first get them here. Letitia Chitoke. Yes, Letitia. Letitia, you can ask your question. I think Letitia's network is not working. Okay, let me mute. So we can continue. Xylem vessels and xylem tracheids are specialized. For a cell to be highly specialized, it means it is kind of modified differently from the normal ones. So first of all, xylem cells are dead. Something dead, it means it will not contain the protoplast. Protoplasts are living contents within the cell. So if a cell is dead, so we don't expect to find their protoplasts. They are long and cylindrical. Their cross walls are perforated or completely absent and form continuous tubes from roots to leaves. And as well, xylem vessels have thick walls made up of lignin. So this is just a general description of them. And there, I would have talked more about these thickenings, but you say that this thing is much covered under histology, but many notes and many people cover them under the xylem tissue. Actually, there's even yes. So xylem, we have this spiral. We already looked at it. Spiral thickenings, which can either be single or multiple. We have annular vessels, annular thickenings. We have reticulate. We have pitted. So these ones, we have already looked at them. And then we can have also differences between the xylem vessels as well as the tracheids. We can have differences between the xylem vessels as well as the tracheids. When you're looking at these, I got this picture from Function Approach by M.B.V. Roberts. This is a common textbook that almost everyone has. And then again, also this picture is from Function Approach. So you can see that the tracheids is tapering. You can see tapering, and it has perforated end walls. Perforations means of tuli. You can see them. And here they had cut it like this so that you can see that inside it is hollow. And then it has lignified walls strengthened with lignin. You can see that they are lignified. And so you can see these are open-ended. But for the case of the vessel elements, take note of this. Some are open-ended, like there is nothing like a barrier, but some could have perforations. They could have perforations in the end walls, like you can see here. Okay, so you must take note of that. So the main conducting cells of angiosperms, and then these ones are the main conducting cells of gymnosperms and fans. For you to understand angiosperms and fans, you must have done a topic called classification. Now, under classification, you can know that we have what we call flowering plants, and there are some that are just vascular, that do not produce flowers. Okay, the non-flowering plants. So I won't go so much into explaining what angiosperm and gymnosperm is. Then short cells are arranged in rows, forming large tubes. Yeah, those are the large tubes. Then these ones are long with overlapping ends. Overlapping means, this is what it means, that if this was, okay, let me redraw it here. I'm assuming if this was the tracheid, like this, okay, it overlaps with another cell like that, like overlapping. In Luganda, it can be like zayingiram, like you can see that this cell would stop here, okay, and this cell would stop here, but they overlap, meaning that this one extends almost into the other like that. That's what we mean by overlapping. But if you check these ones overlap. Like the moment this cell is ending here, that's where it ends, then another one begins. It continues like that, like that, like that. Okay. Then these ones are large inner diameter. These ones are large, of course, the vessel elements, and these ones have a smaller diameter. Then small and abundant in pits, and these ones large and less abundant pits. These ones here. Then perforated plates, no perforated plates. Here, once you enter here, okay, let me first pick some question. Teacher, the question given, give the evidence to support the view that xylem carries water up the stem. Teacher, the question, oh, okay. You're starting, give the evidence. So your question is, give the evidence to support the view that xylem carries water up the stem. Now, those evidences are many, and you can actually find some in understanding biology. They give such evidences, as well as Simpkins, like what can you use to support that xylem carries water up the plant? Okay, so that means I don't know whether I should answer this question now. Okay, for some of the questions which keep on confusing you that are not of the subtopic that we'll be covering, you can always note them down, then maybe we shall begin leaving some minutes to discuss about them. In case a question is not direct to a given subtopic, okay? And then this one, higher efficiency in water conduction, and then this is low efficiency. So the kind of many, this is about describing what you see in the picture, describing see in the picture. Of course, it was looking at describing this vessel element here. This is the slide which I was talking about. Now, this was UNEB 2016 paper 2, number 5. The first question was saying, using illustrations, describe the different types of thickenings found in the vessels of the xylem. Okay, so here they're talking about vessel elements, the vessels of the xylem. And then part B was explain how material are transported in the xylem. Okay, so how can you go about such a question? Being able to explain how materials are transported in the xylem. Then part C was explain the role of transpiration in planning. Now, we have not yet reached here, but we shall talk about this at a later time, about the effect or the role of transpiration in flowering plants. So this one, we have not yet looked at it, but in the notes, it is covered. We shall look at it later when that subtopic reaches. But this one says explain how materials are transported in the xylem. Now this one, you must be able to answer it because we have already covered it, how materials are transported in the xylem, okay? So which theory can you use? Or is it that you are supposed to begin right from the roots, okay? Or you begin direct after water had been taken by from the vascular tissue, from the cortex cells into the vascular tissue. Do you begin from there? And so just explain the cohesion-tension theory. So that's this question indirectly ask you the cohesion-tension theory that explains transpirational pull. I don't know how you understand this question. All you are supposed to begin from the other things down. That's manya in the root hairs. Do you begin from the root hairs or you begin explaining the cohesion-tension theory? But with this question, using illustrations, describe the different types. Okay, let me first see some people contribution. Namaganda Brivian says that you explain cohesion tension theory. Okay, thank you for that. Okay, so such questions are about understanding. I must be able to write. Paper 2 is about writing. Just like this question of Letitia Chitoke, saying that give the evidence to support the view that xylem carries water up the plant. I don't know whether you guys have started synthesizing such a question. Bernard, yes, you have a question.
Yes, I think there is this question, that question of explain how materials are transported in plants, in the xylem. Sorry, I think we describe right from the root hairs.
Okay, that is what you think, eh?
Yes.
Okay, thank you, Bernard, for that contribution. So Bernard thinks that when they tell you to explain how materials are transported in the xylem, then you begin right from the root hairs of uptake. First of all, how water is taken up. So what do you think? Tumwine says root hairs cause it all has, I think you meant that it also has a xylem, okay? And then Sandra says, I think explain the cohesion-tension theory. And then Tumwine, okay, so I think you've seen people's contributions. Letitia says from the root hairs, Okay, now let me end with this Emerald. Yes, Emerald.
So actually, I think that we mainly explain the cohesion-tension theory because mainly transport is accounted by the two theories, cohesion-tension theory and then pressure theory. So for this case, I think you explained the cohesion-tension theory.
Okay, thank you very much, Emerald. Thank you. Let me pick someone else here. Mildred Tranila says cohesion-tension theory. Let me, Tumwine, yes, Tumwine.
So, me, what I think is that we explain right from the root hairs because the xylem, if they have just said transport in the xylem, meaning you explain the whole process of this, of how water moves as long as it is in the xylem up to when it gets out of the xylem. That's what I think.
Okay. Thank you. Yes, Tumwine, that is actually right. Okay, I think this time up. I've just been notified by the admin. Is time up? Someone is saying WhatsApp number. The number is always there on what? In the group. If you are not in the group, the numbers are always there. Okay, in conclusion, because of time, I remember the question is explain how materials are transported in the xylem, okay? So this is going to mainly include that cohesion-tension theory. However, paper 2, let me tell you guys, they never limit you and they will not penalize you. If you have not gotten it from where to begin from, write everything. First, when we are marking, we shall choose the right things. You write everything, you write the biology there, okay? So don't get scared to write. Paper 2, never get scared to write all the biology that you have. Otherwise, enjoy yourselves and goodbye, everyone.